Welcome back, biologists, to your next screencast. Today we're going to talk about Chapter 11, which focuses on single-gene inheritance and meiosis. This chapter's case study focuses on the story of Emily Schaller. She's the second person from the right here. Emily suffers from cystic fibrosis, a condition inherited from your parents. We'll talk about cystic fibrosis, how it's inherited, and how it's treated. Cystic fibrosis can be referred to as CF. Cystic fibrosis is a genetic disease. Once again, it's inherited from your parents. Cystic fibrosis is caused by a single gene mutation. Remember that a mutation is any change to the nucleotide sequence of DNA. When a gene is mutated, it creates a new version of the gene. Any version of a gene is called an allele. A mutant allele for the CFTR gene causes cystic fibrosis. The diagram shows how the deletion of 3 nucleotides from the CFTR gene causes cystic fibrosis. The loss of 3 nucleotides causes a loss of 1 amino acid. The loss of 1 amino acid is enough to trigger cystic fibrosis. Just to review, a gene is a section of DNA on a chromosome that codes for a specific gene or trait. Different versions of that gene are called alleles. Examples of alleles for eye color and hair color are shown here. This graph traces the development of treatments for cystic fibrosis. It also shows that as these treatments have been developed, the median survival age has increased. Today, the median survival age of someone with cystic fibrosis who survives into adulthood is 44 years. This diagram shows that a mutation in the CFTR gene causes the production of faulty CFTR proteins. Because the CFTR proteins don't work properly, salt and water cannot flow well into and out of cells. The ultimate result is an abnormally sticky mucus in the respiratory linings and intestines. This diagram shows many of the symptoms associated with cystic fibrosis. The most important one here is frequent lung infections. These infections can cause lung damage over time, which can eventually lead to death. The CFTR gene codes for the CFTR protein. CFTR stands for Cystic Fibrosis Transmembrane Regulator. This protein acts like a transport protein and helps ions move in and out of cells. The infographic on this slide reviews how the mutation in the CFTR gene produces a faulty CFTR protein. Genes are sections of DNA on chromosomes that provide instructions for making proteins. In our example, the CFTR gene provides the instructions for making the CFTR protein. Remember that humans have about 10,000 genes in their genome. Genes are physically transmitted from parent to offspring during reproduction. The diagram here shows how chromosomes, genes, and DNA are all related to one another. Chromosomes are all made up of genes. Chromosomes and genes are all made up of DNA. Parents pass their genes to their offspring. By the end of this chapter, we want to understand how this happens and how it leads to what we see when we look at someone. Humans are called diploid organisms because we have 2 copies of every chromosome and therefore every gene in our genome. Matching chromosomes are called homologous chromosomes. 1 chromosome in each matching pair comes from our mother. The other chromosome in each pair comes from our father. Human cells have 46 chromosomes, or 23 pairs. The chromosomes within each pair are described as homologous. Because we have 2 copies of every chromosome, which we call homologous or homologs, we have 2 copies of every gene as well. Each copy of the gene will serve the same function, but it may have DNA sequences that differ from one another. If the DNA sequence differs, then we have two alleles of the same gene. In our example, we have the CFTR gene located on chromosome 7. There's a normal version or allele of the gene, and there's a mutant version or allele of the gene. The mutant allele causes cystic fibrosis. One way in which we can look at genes is by studying genotype and phenotype. Genotype is the genetic or allele makeup of an individual. Phenotype is the observable or measurable effect or features of an individual. In the diagrams on this slide, genotype can produce brown mice, or it can produce yellow or green peas. The way in which humans reproduce is called sexual reproduction. It involves the combination of chromosomes, genes, or alleles from a mother and a father. Maternal and paternal genetic material join during fertilization, which is the union of a sperm cell and an egg cell. Maternal and paternal alleles will determine genotype, which will in turn contribute to phenotype, or what we can see or measure. This is an electron micrograph of human sperm cells trying to fertilize an egg cell. Sexual reproduction requires special cells called sperm and egg cells. Sperm and egg cells are more generally referred to as sex cells or gametes. Gametes only carry one copy of each chromosome. As a result, they're called haploid. If we use the letter N to represent a set of chromosomes, haploid cells are N and diploid cells are 2N. We need a special type of cell division to produce haploid or N gametes from normal diploid or 2N cells. This process is called meiosis. In meiosis, we'll take cells that have 46 chromosomes and produce gametes that have 23 chromosomes. These gametes will be genetically unique from one another. This is how we create variety in our offspring. Sperm and egg cells are haploid, meaning they only have one set of chromosomes, or 23 in total. During fertilization, a haploid sperm cell fertilizes a haploid egg cell and becomes a diploid zygote, or fertilized egg. The zygote will divide by mitosis to become an embryo. The embryo further divides and develops to become a fetus and eventually a child. Meiosis can be broken down into two separate divisions called meiosis I and meiosis II. In meiosis I, homologous chromosomes will separate into two daughter cells. Each daughter cell will be haploid, and each daughter cell will have chromosomes that are each made up of two sister chromatids. Try to see and understand this in the diagram on this slide. Meiosis I can be broken down into prophase I, metaphase I, anaphase I, telophase I, and cytokinesis. Let's highlight the important differences. In prophase I, chromosomes will form pairs. In metaphase I, chromosomes will line up in pairs. In anaphase I, the pairs separate versus sister chromatids separating. In telophase I and cytokinesis, you end up with two daughter cells that are haploid or have half the normal number of chromosomes. In meiosis II, sister chromatids will now separate. This will result at the end in 4 haploid daughter cells. These daughter cells will eventually develop into sperm and egg cells. Meiosis II can be broken down into prophase II, metaphase II, anaphase II, telophase II, and cytokinesis. Meiosis II looks a lot like mitosis. During metaphase II, chromosomes line up single file. In anaphase II, sister chromatids separate from one another.